Okay, so let us start the class. Today I will discuss about the digestion and metabolism of protein in ruminant and non-ruminant species. So the digestion of protein, which mainly happens in stomach and a small intestine, the upper part of the small intestine, duodenum, by the action of different enzymes. I am talking about the digestion of protein, digestion and absorption of protein in monogastric animal first, monogastric species, and the different enzymes like pepsin, which is being secreted from the stomach, and trypsin, chymotrypsin secreted from the pancreas, and breakdown of the food protein into polypeptides, and finally into the amino acids. So in that way, the digestion and absorption of the protein takes place. And the gastric phase of digestion, there are two phases. Later on, I will discuss the gastric phase. And so in gastric phase of digestion, the secretion of pepsinogen takes place from the chief cell. Chief cell and HCl was secreted from the parietal cell. So two types of the cells present, chief cell and parietal cells, which are responsible for the secretion of pepsinogen and HCl. So during gastric phase of digestion, the pepsin and HCl, which are principally responsible for gastric digestion, and pepsinogen, which is generally in inactive form. And inactive form of pepsin is, we can say that the inactive form of pepsin is pepsinogen, and HCl activates the pepsinogen. HCl activates the pepsinogen, which are converted into pepsin. So by the activation of the HCl, the pepsinogen is converted to pepsin, and further hydrolysis, which hydrolyzes the peptide bonds. So pepsin, which has a strong clotting action, you are aware about that. The pepsin has a strong clotting action on milk and renin or chymosin, which are enzymes that occurs in gastric juice of calf and young piglets. So these renin and or chymosin occurs in gastric juice of young piglet or calves. Calves means before development of the rumen, these enzymes are being present, which is responsible for and which has a strong clotting action of the milk. And the product of protein digestion in the stomach, which are principally polypeptides of different chain or variable chain, we can say that variable chain length, and few amino acids also. And the histamine, you are aware about you must have heard the name of the histamine, which starts releasing in response to vagus nerve stimulation. And secretin hormone, secretin, which prevents the acid secretion, but stimulates the pepsin secretions. And similarly, the gastric inhibitory polypeptides also inhibits pepsin secretions. So now coming to the digestion of protein in a small intestine, how it can happen. Generally, the majority of digestion and absorption happens in a small intestine, and primary site is duodenum and jejunum. And the intestinal digestion, which comprises of intraluminal digestion, and membrane and intracellular digestion. So under intraluminal digestion, what happens? The digestion is mostly performed by pancreatic enzymes. I will show you one table where all the enzymes and site of secretions are present or depicted there. So you have gone through this table. Then you may see that from where the enzymes are being secreted and what is the action, mode of action or action of the enzymes and what it will, how it will happen in the intestine. So a stimulation of pancreas by vagus nerve, which results in secretion of fluids, bicarbonates, and different pancreatic enzymes. And peptide hormones like secretin, which releases in response to acids, which stimulates the secretion of fluid and bicarbonates. And cholecystokinin, another hormones, cholecystokinin, released in response to the presence of food, and which stimulates the secretion of pancreatic enzyme as well. So in membrane and intracellular digestion, what happens? The final stage of protein digestion. It's the final stage of protein digestion at external membrane of intestinal mucosa, also known as crust border cells, within the small intestine, columnar cells. So this table, I was talking about this table, the some important digestive enzymes of proteins. So different enzymes in one column, systemic name, in another column, source of secretion, and substrate where they acted upon. So this table is also available in the McDonald book. You can refer this book also to see these tables. I will share this PPT in the groups. So you go through this table. So now coming to the absorption of protein. Absorption of the protein that is very much important, and the digested product of the protein, which is available in the lumen of intestine, are free amino acids. And there are some small oligopeptides also available there. And further, they enter the epithelial cells of a small intestine and hydrolyzes by a specific di or tripeptidases. Di or tripeptidases, where some small peptides are absorbed intact and subsequently it appears in the portal circulation. So liver play a very important role in the absorption of the protein, and amino acids which subsequently pass into the portal blood and then into the liver, and absorption and the absorbed amino acids from the small intestine happens by active transport mechanism, or active transport mechanism, also we can say that sodium dependent mechanism, except you must remember the name of the amino acid, except glycine, proline, and lysine. Except glycine, proline, and lysine. And the rate of absorption of amino acid which fluctuates. So all kind of the amino acid absorption rate is not similar. So rate of absorption, which may fluctuates depends on the types of the amino acid. Like rate of absorption is higher in case of methionine. When we compare about the methionine to valine. So rate of absorption of valine is poor than the methionine. Now coming to the digestion and absorption of protein in ruminants. So in case of the ruminant, the digestion of protein, digestion and absorption is different from the non-ruminant. So dietary protein, which are being hydrolyzed to peptides and amino acids by rumen microbes. So there are different kind of the rumen microbes which are responsible for the proteolysis of the dietary protein, and the two primary microbes which are responsible for that and have proteolytic activity, that is Parvotella, Ruminocolla, and Peptostreptococci species. And protozoa is also playing a very important role in digestion and absorption of the protein. In case of ruminant, and you remember that per kilogram of organic matter digested, in general, I am talking about in general, the per kilogram of organic matter digested in most of the feed in the rumen, which yields approximately 200 grams of microbial protein. So per kg organic matter will yield, in case of ruminant, digestion of the per kg organic matter yields 200 gram of microbial protein. But in case of when we offered rapidly fermented food or immature grasses or fodder crops, which having high amount of the soluble carbohydrates, in that case, the microbial protein yield will be higher. That is 260 gram per kg organic matter digested. So 60 gram higher microbial protein will be produced when we are offering highly soluble carbohydrate or lush green pasture to the animals. So you also remember these two microbes, name of these two microbes, Parvotella, Ruminocolla, and Peptostreptococci species, and Protozoa as well. So these organisms are mainly responsible for proteolysis and having activity of proteolytic activity. So when rumen microbes pass through the abomasum and a small intestine, their cell protein get digested and absorbed. So there are two types of the protein present in the diet. That is rumen degradable protein and rumen undegradable protein. In this figure, you can see that when we offer the diet to the animals, so 40% of the protein in the most of the diet is undegradable in nature, and that will escape the rumen and escape the fermentation process and goes to the lower tract, get digested and absorbed into the system. But 60% they could not escape, they are coming under the rumen degradable protein, RDP in nature, and then microbial action after that dietary protein takes place and release of the ammonia. So during fermentation process, ammonia will be released, and then this ammonia will be utilized by the different microbes for as a source of nitrogen, and by that way these microbes will get multiplied and multiply their number. So nitrogen, so as a nitrogen source, these microbes are being dependent on the dietary source of the protein to get the nitrogen. And excess ammonia will be excreted through urine and other sources, and some of them goes to the liver and then recycled by the saliva. So further, the nitrogen present in the saliva again comes to the rumen, and then this cycle will continue. So this is the continuous process, and by that way, the dietary protein is being utilized in presence of fermentable energy sources, and there are conversion of the different amino acids. It depends on the diet, which kind of the diet you are offering to the animals. So rumen bacteria, it is not happens in the non-ruminants. So rumen bacteria are capable of synthesizing dispensable as well as indispensable amino acids. So there is no provision of dietary requirement of essential amino acid in ruminant. But in case of high yield animals, we can supplement some essential amino acids to the animals to maintain the production performance of animals. But in generally, the ruminant animal could not require, or we cannot offer essential amino acids to ruminant because they have capability to synthesize both essential and non-essential amino acids with the help of rumen microbes. So ammonia in rumen liquor, which is the key intermediates in microbial degradation and synthesis of the protein, and the ideal concentration of ammonia. If ammonia is in excess quantity, then what happens? Ammonia toxicity will take place. So we should remember there is ideal condition, and we should maintain the ideal condition of the ammonia in the rumen. Otherwise, toxicity will happen. So concentration of ammonia in rumen liquor, which vary from 85 milligram per liter to 300 milligram per liter of rumen liquor. These data are also available in the books, so you can see these data in the book also, and try to remember the data. What is the optimum concentration of the ammonia in rumen liquor and ammonia in blood as well. So there are different factors which affect the protein digestibility, like enzyme inhibitors, some enzymes inhibitors which are present in the diet itself, like trypsin inhibitor, which is present in the soybean. So these inhibitors which affect the digestibility and degradability of the protein in the rumen. Heat damage protein, when we treated the grains and protein rich grains like soybean by heating process, that is one of the method of physical method of treatment or protection of the protein. During protection process, or when we making the protein undegradable, then sometimes excess heating of the grains that restrict the utilization of the nitrogen present in the particular grain. So that reaction is called Maillard reaction or Browning reaction. So what is Maillard reaction or Browning reaction? Both are same, Browning reaction. And how the changes, what kind of the changes occur during the Maillard reaction, during treatment or heat treatment of the grain, you must know about that, what will happen, because short notes always asking by external examiners or internal also what is Maillard reaction. So you should know about that. So what happens in reaction, in this reaction, the primary amines of peptide chain, that is the amino group of lysyl residue, and the reducing sugars such as xylose, which are most reactive sugars, xylose, glucose, and galactose, and forms an amino sugar that forms amino sugar complex by isomerization process and polymerization. And that trypsin, which cannot work on this lysyl bond, thus reducing the availability of amino acids or protein availability, where the reaction which are affected by the sugar concentration, duration of heating is also very much important. कितने समय तक उसको हीट किया, ज्यादा समय तक करेंगे तो ज्यादा इरिवर्सिबल कॉम्प्लेक्स बन जाएगा फिर, ठीक है. So sugar concentration depends on sugar concentration, duration of heating, water activity, pH of reactions. So these are the factors which affect the Browning reactions or formation of Maillard reactions. Next factor is the source of protein, which also affect the digestibility. So source is also very much important. Which source we are offering to the animals is very much important. That will affect the digestibility of the protein. And amount of the fiber present in the diet. So fiber availability in the diet also affect the digestibility of the protein, and the digestive enzymes present in the animals. So it also depends on the species of animal and the rumen size and rumen development, rumen papilla development. So a species also one of the factor. In some species, more digestive enzymes are being produced. So it depends on the types of the animals. So digestive enzymes, the development of the animals is also one of the important factors which affect the digestibility of the dietary protein. Now coming to the metabolism of the protein, how it takes place. There are two important processes that are compulsory to synthesize dispensable amino acid and oxidize excess amino acid, as well as for gluconeogenesis process, that is transamination and deamination process. So there continuous synthesis and breakdown of the protein takes place inside the system. And what does it? It refers to protein turnover. So protein turnover is continuous synthesis and breakdown of the protein in the system. And liver is acting as a buffer or focal point, we can say. Liver is acting as focal point of regulating the protein metabolism. So protein metabolism can be essentially the liver is focal point for regulating of protein metabolism, and transamination and deamination process, which is compulsorily required for metabolism of the protein. So what is that? So coming one by one, what is transamination and what is deamination? So transamination, it refers to the exchange of ammonia between amino acid and alpha keto acid. This is reversible type of the reaction, and this reaction occurs in mitochondria and cytoplasm. So the transamination process occurs in mitochondria and cytoplasm of the most of the cells, in some cells maybe differs, but most of the cells it occurs in mitochondria and cytoplasm. And vitamin B6, which is essential component of these reactions. You can see in this diagram how it takes place. This is the fate of absorbed amino acid. So transamination and oxidative deamination process. Actually, deamination is oxidative and non-oxidative type. So transamination and oxidative deamination process, how it will happen. So deamination, what is that? Deamination, there are two major type of deamination process, such as oxidative and non-oxidative type. So in case of oxidative deamination, the amino group of most of the amino acids, the amino group of most of the amino acids are transferred to ketoglutarate by transamination process. And the release of ammonia is catalyzed by alpha glutamate dehydrogenase. In this diagram, you can see that alpha glutamate dehydrogenase, and the fate of end product is ammonia to urea cycle, and alpha ketoglutarate to TCA cycles. In case of non-oxidative deamination, which requires amino acid dehydratase and vitamin B6. And pyridoxal phosphate is active form of vitamin B6. You know about that. And there are wide range of enzymes which depends on vitamin B6, like transaminase, synthetase, decarboxylase, dehydratase, etc. for amino acid metabolism. So different enzymes are responsible for amino acid metabolism and its absorption. So now coming to the what is the fate of absorbed amino acid in the system. Then after I will come to the protein synthesis. Actually, there are three processes of protein synthesis: first initiation process, then elongation process, then termination process. So we should not go in details, just process. You might have remembered that there are three processes of protein synthesis: initiation, elongation, and termination process. And detail you might have read in biochemistry classes. So what will happen? The fate of absorbed amino acid, what will happen? That when amino acids consumed in excess amount, जब ज़्यादा amount में amino acid or through diet you may consume, then what will happen? The required for protein synthesis, then it cannot be stored. So excess amino acid will be excreted from the body. So there is wastage of amino acid and wastage of the money as well. So whatever the required for the animals, we can offer only those or fulfill only those requirements. That will be the best idea. Otherwise, it will excreted from the body. And you might have heard the name of the anabolism and catabolism process. The two processes which take place: anabolism, which represents protein synthesis. That means muscle protein, milk protein, egg protein, repair and maintenance of the cells of the body. So by process of anabolism, protein synthesis takes place. While in catabolism, which involves broken down of the protein. So broken down of the protein and used as a source of energy by gluconeogenesis or nitrogen for urea synthesis. So protein synthesis from the amino acids, which becomes either from the in process of the digestion, and there are three basic process of the protein synthesis: initiation, elongation, and termination. So you might have remember the name, this process, and what is the significance of the protein turnover? So significance of protein turnover or metabolism, that plays is very important role in metabolic adaptation, like enzymes, hormones. So these are the significance, and which accelerates restructuring of growth and other activities like muscle synthesis, collagen synthesis. It also helps in repairing of the cells and tissues. So very much important for repairing of cells and different body tissues. It also plays very important role or crucial role, we can say, in mobilization or transport of the protein during protein deficiency condition. So, and it can be used as source of energy. So gluconeogenesis, by gluconeogenesis process, it can be used as a source of energy as well. And for removal of the faulty molecules or proteins from the system. So these are the significance of the protein metabolism. Now coming to the factors which influencing the body protein turnover or metabolism. What are those factors? So you should remember these factors as well. And like first factor is the genetic variations. Genetic variation in respect of the species, in respect of the breeds, in respect of the different strains of bird, when we can say that. So these are the factors which may affect or influence the body protein metabolism or turnover. Second factor is nutrition. So nutrition means quality and quantity of the protein, which is present in the, which also depends on the types of the diet and quality, which quality diet or which quality ingredient offering to the animals, and how much quantity you are offering to the animals. And that diet, how much amount of the protein, which kind of the protein is produced. So energy source of the diet will also influence the metabolism of the protein. Physiological status of the animal, like productive status, unproductive status, which condition the animal are, lactating a stage,